So 12th class, now we are going to study the next chapter, that is chapter 11, Biotechnology Principles and Processes. Before starting the chapter, I will discuss some learning outcomes with you, that what things you will get to know after completion of this chapter. So learning outcomes of this biotechnology is that you will come to know about the techniques that are used in the processing of biotechnology. You will become aware about the principles that are employed in biotechnology. You will understand the procedure of PCR and how does it work. You will come to know about the various tools that are used in biotechnology, as well as last point, that is the downstream processing. So how these things work, we will discuss these one by one. So before starting the chapter, that is biotechnology principle and processes, first of all you should be clear that what is the meaning of biotechnology. As you can see, the biotechnology, it means that the various techniques that are you using in agriculture, in food processing industries, medicine diagnostic, bioremediation, waste treatment and energy production. Now before moving further, first of all you should be clear that what is the meaning of the this bioremediation. Actually the bioremediation means these are the techniques by which we reduce the pollution level of a particular area. Means how can we reduce the polluting potential of an area, that is studied under bioremediation. Waste treatment, as you know that the microbes they are used in the treatment of waste material, that is the sewage treatment. As well as energy production, as you can say that in biogas production the methanogens will be used and they will result in production of energy that can be utilized as a source of energy. Similarly, in the various food processing industry, now what does it mean that how biotechnology that is used in food processing industry? All of you know that the microbes they are being both harmful as well as useful. But if you come to know that what is the importance of these microbes in your daily life, then there are many examples that you can easily explain. The most common example that is I think that is used by everyone in their houses. For example, the preparation of the curd that is used in each and every home. So in that the lactobacillus bacteria they are playing an important role. Even in the medicine field you can say that the antibiotics which are used for treatment of many diseases, all these are due to the microbes. So how these things, or you can say how these antibiotics are prepared, how on the commercial level this curd, this cheese, as well as you can say the alcoholic drinks, how all these things are produced, so all that will be discussed in this chapter. Now moving further, EFB, as you can see that it is the EFB. What does EFB means? EFB is actually the European Federation of Biotechnology. They had provided the definition of biotechnology. What they said, that it is the integration of natural science and organism, cells, parts thereof and molecular analogues for the product and services. What is the meaning of this definition? That biotechnology is the branch of science that is involving the integration of science and organisms. Organism means the microbes. It means that microbes and the natural science both are integrated with each other. Their cells or their parts may be used and they are resulting in the production of the various products. So all this will be included in the biotechnology. Now moving further, what are the bases of modern technology? Bases of modern technology means what principles that are used in the modern technology. The first one is the genetic engineering. As you can see, it is the genetic engineering, as the name itself indicate that engineering in the field of genetics. Genetics means the branch of the science that deals with the study of genes, that all will be studied in the genetics. So if the engineering is done at the genetics level, means at the gene level, then you will say it is the genetic engineering. Now what is being done in genetic engineering? discussing further, as you know that DNA or RNA that act as the genetic material in most of the organisms. DNA is the genetic material. So if we change the DNA or RNA of an organism, so obviously it will affect its phenotype. All of you know that phenotype is the external appearance of an organism. So obviously if the DNA or RNA of an organism is changed, it will affect its phenotype. Second thing, aseptic technique. We had altered the DNA or you can say the RNA of the particular host, then it's necessarily not that it will result in the alteration of its phenotype. Further, we need aseptic technique. Aseptic technique means these are the contamination free condition. Aseptic condition means if you had altered the genetic material, further we will need a contamination free condition so that the alteration which we had done that can show its effect, right? So for the manufacturing of the products such as antibiotic, vaccines, etc., we need to maintain a proper aseptic conditions. And why there is the requirement of aseptic condition? This is done so that the growth of only the desired microbes can take place, because if the undesired microbes will grow, then it will result in affect the process. It will not take place in a proper way. Now the first thing is the genetic engineering. Before starting the genetic engineering, we know that there are two types of reproduction. One is asexual reproduction and second is the sexual reproduction. What is the difference between these two? Obviously, the asexual reproduction that does not create variation. It means that it will preserve the genetic information, while the sexual reproduction preserve the variation. This is the fundamental difference between these two. And the conventional method of hybridization. Hybridization means in which we combine the two things and they result in the production of new thing. But what happens in case of commercial hybridization? In commercial hybridization, we need only the desired one. But in commercial hybridization, it doesn't matter. Along with the desired gene, undesired genes are also multiplied. This is the major disadvantage of conventional hybridization. That's why this has to be removed. This disadvantage have to be changed. And for that, there is the process that is known as genetic engineering. Genetic engineering involves the selection of only the desired gene. It does not involve the use of any undesired gene. So there is no chance that undesirable genes, they will also multiply with desired genes. So genetic engineering had overcomes this limitation. Now this genetic engineering includes three major steps, as you can see on the screen. First one is the creation of the recombinant DNA. Recombinant DNA, the DNA which is formed by combining two DNA. Second is the gene cloning, that is the multiplication of that DNA. Third one is the gene transfer into the host organism. In third step, the gene will be transferred into the host organism. So these three steps, these are the basic in the process of genetic engineering. Now see, the piece of the DNA that we want to introduce in the host, it is not capable of replicating itself in the host. But if it is the part of the host DNA, then it can easily multiply. So what we have to do in this case, we need a vector. As you know the meaning of vector, even in the case of malaria, you can say that the female mosquito, female anopheles mosquito that carries the plasmodium from the infected person to the healthy person. In the same way, this vector that are used in the process of genetic engineering, that also carry the foreign DNA into the host, means these vector will transfer the foreign DNA piece into the host cell so that it can multiply. And when the foreign piece of DNA had entered into the host DNA, it will start multiplying, and this process is known as cloning. As you can see, what is the meaning of cloning? The process of formation of multiple identities, copies of DNA is known as cloning. So you will get multiple copies of the gene of interest. Gene of interest means the piece of DNA that you will introduce in the host.